Chapter 26 on nucleic acids and protein synthesis. This is the first part really focusing on the structure of nucleic acids. So just here to summarize and make you understand what are DNA, chromosome, and genes. Well, if you look at those figures here on the left side, this is a non-dividing cell. When a cell is not dividing, the nucleus is occupied by what we call chromatin. So if you look at this, it's just very, very loose. Now, when a cell is getting ready for division, the chromatin is going to tangle itself and is going to actually be able to curl itself upon histone. And getting ready for division, you see that eventually you see a visible chromosome, which is here two replicated sister chromatid. So chromatin is a compact, orderly tangle of DNA. DNA stands for deoxyribonucleic acid, and that is what carries the genetic information. It's made of deoxyribonucleotide, and it's twisted around organizing proteins we call histones. During cell division, the chromatin becomes even more compact and is going to turn into what we call chromosomes. Chromosomes are complex of protein and DNA. You can only see them, so they're only visible during the cell division. Now, all of those contain different DNA molecules, and all that DNA has to be duplicated, we say replicated, so each new cell, the daughter cell, will have exactly the same copy. So looking closely at the DNA molecule, it's made of genes. Genes are here, part individual segment of the DNA. They contain all the instructions that are going to be leading to synthesis of polypeptide. But very interesting, not all genes coded for and by an organism are expressed as protein. Some genes code for functional RNA molecule. So looking at here, you see that organisms very widely differ in the number of chromosomes they have. For example, a horse has 64 chromosomes organized in 32 pairs. We're going to explain why the pairs are important. A cat has 38 chromosomes, mosquito 6, a corn plant 20, and human has 46 chromosomes. Please note that the number of chromosomes doesn't make the organism more sophisticated or more advanced. So looking at here the composition of nucleic acid, well, they're the same as carbohydrate or protein. They are polymers. And when we're looking at here, nucleic acid are polynucleotide. Each nucleotide has three parts: a 5-membered cyclic monosaccharide, the sugar, a nitrogenous base, and a phosphate group. So you see here the structure of a nucleotide you should be able to recognize. The nitrogen base on top, a monosaccharide here, deoxyribose, and a phosphate group. We have two types of nucleic acid, the one called DNA and the one called RNA. RNA stands for ribonucleic acids. We find them in different kind of forms. Some of them are called messenger RNA, the other are transfer RNA, and the other one are ribosomal RNAs. Looking at the sugar, well, difference, extremely important difference between DNA and RNA is the sugar part of the molecule. RNA, the sugar is a ribose, and that's where you find in ribonucleic acid. In DNA, the sugar is a deoxyribose, so deoxyribonucleic acid. So deoxyribose, deoxy means that you're missing an oxygen from the carbon number 2 of the ribose. So looking here on the left, that's the RNA. On the right side, the DNA deoxyribose is missing an oxygen. Next is looking at the bases, where there are 5 nitrogenous bases. Some of them are belonging to purines, other pyrimidine. For every base, we have a 1-letter code. A stands for adenine, G for guanine, C cytosine, T thymine, and U uracil. Uracil is only found in RNA, and adenine, guanine, cytosine are found in both DNA and RNA. So looking at here the composition still, the molecule with the ribose or deoxyribose and one of the 5 nitrogenous bases, we call that a nucleoside. So nucleoside, the sugar, the base, and we look at here the name is modified by the suffix eosine for the purine base and iodine for the pyrimidine base. Deoxy is for the one who have deoxyribose. Then a little bit more complex, where we get to a nucleoside plus a phosphate, that becomes a nucleotide. They are the monomers of both DNA and RNA. Now, we're going to look at the numbering of the carbon atom. So nucleotide is a 5' monophosphate ester of a nucleoside, and we name them by adding 5' monophosphate at the end of the nucleoside name. Now, looking at here, we make the distinction between the atom in the sugar ring from the one in the base rings. Numbers without primes are used for the atom in the base, and numbers with primes are used for the atoms in the sugar. So looking at here on the left, deoxyribonucleoside, you see that here the nitrogen, that's the base, and then you see on the sugar the 1', 2', 3', 4', 5'. So that tells us here that the phosphate group will be added to the 5' carbon atom that you have in the sugar. So summary, nucleoside is the sugar and the base. The nucleotide is the sugar, the base, and the phosphate group. Now again, DNA stands for deoxyribonucleic acid. The sugar is the 2-deoxyribose or D-ribose, and the base is adenine, guanine, cytosine, thymine. RNA, they are ribonucleotide with a D-ribose, and the bases adenine, guanine, cytosine as the same as for DNA, but then thymine is replaced by uracil.